17 shows the general principle of embodied motion. Basically, these result from the continual formation of new cell membrane at leading edge of the pseudo- pseudopodium and continual absorption of the membrane in the mid and rear portion of the cell. Two other effects are also essential for forward movement of the cell. First is attached of the pseudopodium to surrounding tissue so that it becomes fixed in its leading position, while the remainder of the cell body is being pulled forward toward the point of attachment. This attachment is caused by the receptor proteins that line the inside of the exocytic vesicles. When the vesicles become part of the pseudopodial membrane, they open so that they are insides event of the outside, and the receptors now protrude to the outside and attach to ligands in the surrounding tissue. At the opposite end of the cell, the receptor pull away from the ligands and from new endocytic vesicles. Then the inside the cell, these vesicles stream toward the pseudopodial end of the cell, where they are used to form new membrane for the pseudopodium. The second essential effect for locomotion is to provide the energy required to pull the cell body in the direction of the pseudopodium. A moderate to large amount of the protein actin to the cytoplasm of the cells. Much of the actin of the from actins is in the form of single molecules that do not provide any motive power. However, these molecules polymerize to form a filamentous network, and the network contacts with it binds with the actin binding proteins such as the myosin. The entire process is energized by the high energy compound ATP. This is what occurs in the pseudopodium of the moving cell, where such a network of action filaments forms a new inside the enlarging pseudopodium. Contraction also occurs in endoplasm of the cell body, where pre-existing actin network is already present beneath the cell membrane. Type of cell that exhibit the amoeboid locomotion.
The most common cell to exhibit amoeboid locomotion in the human body are the white blood cell when they move out of the body into the tissue to form tissue macrophage. Transfer RNA, the anticodons. Type Another type of RNA that is essential for protein synthesis is called transfer RNA because it transfers amino acids to protein molecules as the protein is being synthesized to protein molecules as the protein in being synthesized. Each type of tRNA combines specifically with one of the 10 amino acids that are to be incorporated into proteins. The tRNA then acts as a carrier to transport its proteins. The tRNA then acts as a carrier to transport its specific type of amino acids to the ribosomes where protein molecules are forming. In the ribosomes, each specific type of tRNA recognized a particular codon on the mRNA described later and thereby delivers the appropriate amino acid to the appropriate place in the chain of the newly forming protein molecule. Transfer RNA, which contains only about 80 nucleotides, is a relatively small molecule in comparison with mRNA. It is a folded chain of nucleosides with a clove leaf appearance similar to that's shown in figure 3 to 9. At one end of the molecule, there is always an adenylic acid, which the transported amino acids attach to a hydroxyl group of the ribose in the adenylic acid. Because the function of tRNA is to cause attachment of a specific amino acid to a forming protein chain, it is essential that each type of tRNA have specify for a particular codon in the mRNA. The specific codon in the tRNA that allows it to recognize a specific codon. A specific codon is again a triplet of nucleotide because it is called an anticodon. This anticodon is located approximately in the middle of the tRNA molecule at the bottom of the clover leaf configuration shown in figure 3 to 9. During formation of the protein molecule, the anticodon bases combine loosely by hydrogen bonds with the codon base of the mRNA. In this way, the respective amino acids are lined up one after another along the mRNA chain, thus establishing the appropriate sequence of amino acids and newly forming protein molecules. Figure 3 to 9. A messenger RNA sent is moving through ribosomes. As each codon passes through, an amino acid is added to the growing protein chain, which is shown in the right hand ribosome. The transfer RNA molecule transports each specific amino acid to the newly forming protein.
Type of RNA in the cell is ribosomal RNA, which constitutes about 60% of the ribosome. The remainder of the ribosome is protein, including about 75 types of proteins that are both structural proteins and enzyme needed to the manufacture proteins. The ribosome is the physical structure in the cytoplasm on which the proteins are actually synthesized. However, it always functions in association with the other two types of RNA. The transfer RNA transports amino acid to the ribosome for the incorporation into the developing proteins, whereas the messenger RNA provides the information necessary for sequencing the amino acid in proper order for each specific type of protein to be manufactured. Thus, the ribosome acts as a manufacturing ribosome in the nucleus. The DNA genes for the formation of ribosomal RNA are located in 5 pairs of the chromosome in the nucleus. Each of these chromosomes contains many duplicates of these particular genes because of the large amounts of ribosomal RNA required for cellular function. As the ribosomal RNA forms its collects in the nucleus, a specialized structure lying adjacent to the chromosome. When large amounts of ribosomal RNA are being synthesized, as occurs in cells that manufacturing large amounts of protein, the molecules is a large structure, whereas the cells that synthesize little protein, the nucleus may not even be seen. Ribosomal RNA especially proceeds in the nucleus, where it binds with the ribosomal proteins to form the granular condensation products that are the primordial subunits of ribosome. These subunits are then released from of the nucleus and transported through the large pores of the nuclear envelope to almost all parts of the cytoplasm. After the subunits enter the cytoplasm, they are assembled to form the mature functional ribosome. Therefore, the proteins are formed in the cytoplasm of the cell, but not in the cell molecules. Does not contain mature ribosomes.
miRNA and small interfering RNA. A fourth type of RNA is the cell is called microRNA. miRNA are short 21 to 23 nucleotides single standard RNA fragments that regulate gene expression. Figure 3 to 10. The miRNAs are encoded from the transcribed DNA of gene, but they are not translated into proteins and are therefore often called non-coding RNA. The miRNA are processed by the cell into molecules that are complementary to mRNA and act to decrease gene expression. The generation of miRNAs involves special processing of longer primary precursor RNA called pre-miRNA, which are primarily transcriptions of gene. The pre-miRNA are processed in the cell molecules by microprocessor complex to miRNAs, which are 60 nucleotides stem loop structures. These pre-miRNAs are further processed into cytoplasm by a specific dicer enzyme that helps assemble an RNA-induced silencing complex RSIC and generates miRNAs. Complementary region of the mRNA and protein progressions of translation of degradation of the mRNA before it can be translated by the ribosome. miRNA are believed to play an important role in normal regulation of cell fluid. The alteration in miRNA function have been associated with disease such as cancer and heart disease. The association with disease, which is cancer, is heart disease. miRNA. Figure 3 to 10. Regulation of gene expression by microRNA, miRNA. Primary miRNA, pre-RNA. The primary transcriptions of gene processed in the cell nucleus by the microprocessor complex are converted into pre-miRNAs. The pre-miRNAs are then further processed into the cytoplasm by dicer, an enzyme that helps assemble an RNA-induced silencing complex (RSC) and generates miRNAs. The miRNAs regulate gene expression by binding to the complementary region of the RNA and repressing translation or promoting degradation in the messenger RNA before it can be translated by the ribosome.
MicroRNA is a small interfering RNA or siRNA, also called the silencing RNA or short interfering RNA. The siRNAs are short, double-stranded RNA molecules composed of 20 to 25 nucleotides that interfere with the expression of specific genes. siRNAs generally refer to synthetic messenger RNA and can be administered to silence expressed of specific genes. They are designed to avoid nuclear processing by the microprocessor complex, and after this siRNA enters the cytoplasm to activate the RASC silencing complex, blocking the translation of messenger RNA. Because of siRNAs can be tailored for any specific sequence in the gene, they can be used to block the translation of any messenger RNA and therefore the expression by any gene for which the nucleotide sequencing is known. Researchers have proposed that siRNAs may become useful therapeutic tools to silence genes that contribute to the pathophysiology of disease.
Translation formation of proteins on the ribosomes. When a molecule of mRNA comes in contact with a ribosome, it travels through the ribosome beginning at a predetermined end of the RNA molecule specified by an appropriate sequence of RNA base called the chaining codon. Chaining dating codon. Then, as shown in figure 3 to 9, while the mRNA travels through the ribosome, a protein molecule is formed, a process called translation. Thus, the ribosome reads the codon of the mRNA in much the same way that a tape is read as it passes through the playback head of a tape recorder. Then, when a stop or a chain terminating codon slips past the ribosome and the end of protein molecule is signaled, the protein molecule is freed into the cytoplasm. Polyribosome. A single mRNA molecule can form protein molecules in several ribosomes because the initial end of RNA strand can pass to a successive ribosome as it leaves the first. As shown at the molecule are in different stages of development in each ribosome. As a result, clusters of ribosomes frequently occur with 3 to 10 ribosomes being attached to a single mRNA at the same time. These clusters are called polyribosomes. An mRNA can cause formation of protein molecules in any ribosomes. There is no specificity of ribosomes for given types of protein. The ribosomes are simply the physical manufacturing plant in which the chemical reactions take place. Many ribosomes attach to the endoplasmic reticulum. In chapter 2, we noted that many ribosomes become attached to the endoplasmic reticulum. This attachment occurs because the initial end of many forming protein molecules have amino acids sequences that immediately attached to a specific receptor sits on the endoplasmic reticulum, causing these molecules to penetrate the reticulum wall and enter the endoplasmic reticulum matrix. This process gives a granular appearance to the portion of the reticulum where proteins are being formed and are entering the matrix of the reticulum. Figure 3 to 11. The physical structure of the ribosome as well as their functional relationship to messenger RNA transfer DNA and the endoplasmic reticulum during the formation of protein molecules. Figure 3 to 11 shows the
functional
relationship of our ribosome attached to the membrane of the endoplasmic reticulum. Note the process of translation occur in several ribosomes at the same time in response to the name stand of mRNA. Note also the newly forming polypeptide chain passing through the endoplasmic reticulum membrane into the endoplasmic matrix. It should be noted that except the granular cells in which large amount of protein containing secretory vesicles are formed, most proteins synthesized by the ribosomes are released directly into the cytosol instead of into the endoplasmic reticulum. These proteins are enzymes and internal structure proteins of the cell. Chemical steps in protein synthesis.
Some of the chemical events that occur in the synthesis of the protein molecule are shown in the figure 312. This figure shows the representative reaction for three separate amino acids AA1, AA2, AA20. AA1, AA2, A20. The stages of reaction are as follows. Each amino acid is activated by the chemical process in which ATP combines with the amino acid to form the adenosine monophosphate complex with the amino acid, giving up with high energy phosphated amino acid having the excess of energy that ইয়ে কি বলে এই তুমি state amino acid have an excess of energy then combines in each specific transfer RNA to form an amino acid tRNA or transfer RNA complex at the same time realize the adenosine monophosphate. The transfer RNA carrying the amino acid complex then comes in contact with the messenger RNA molecule in the ribosome, where the anticodon of the transfer RNA attaches temporarily to its specific codon of the RNA, messenger RNA, thus lining up the amino acid in the appropriate sequence from a protein molecule. Then under the influence of the energy peptide transferase, one of the proteins in the ribosome, peptide bonds are formed between the successive amino acids, thus adding the progressively to the protein chain. These chemical events requires energy from the additional high energy phosphate bond, making a total of 4 high energy bonds used for each amino acid added to the protein chain. Thus, the synthesis of proteins is one of the most energy consuming process of the cell. Peptide linkage or combination of amino acid. The successive amino acid in the protein chain combine with one another according to the typical reaction. This chemical reaction, a hydrolysis radical, which is removed from the COH protein of the first amino acid and the hydrogen of the NH2 protein of the other amino acid is removed. This combine to form water and the two reactive sites left on the two successive amino acid bond with each other, resulting in a single molecule. Thus, process is called the peptide linkage. As such, the additional amino acid is added and additional peptide linkage is formed. Synthesis of other substance in the cell. Many thousand protein enzymes formed in the manner just described the control is essentially all the other chemical reaction that take place in the cells. These enzymes promote the synthesis of the lipid, glycogen, purines, pyrimidines, and the hundreds of the other substances. We discussed many of these synthetic processes in relation to carbohydrate, lipid, and the protein metabolism in chapter 68 through 70. These substances each contribute to the various functions of the cells. Control of gene function and the biochemical activity in cell. From our discussion thus far, it is clear that the genes control both the physical and chemical function of the cells. However, the degree of the activation of respective genes must also be
controlled, otherwise some parts of the cell might overgrow or some chemical reactions might overreact until they kill the cell. Each cell has powerful internal feedback control mechanism that keeps the various functional operations of the cell in step with another. For each gene, 20,000 to 25,000 genes in all, at least one such feedback mechanism exists. There are basically two methods whereby the biochemical activities in the cell are controlled: genetic regulations, in which the degree of activation of the gene and the formation of gene products are themselves controlled, and two, enzyme regulations, in which the activity levels of already formed enzyme in the cell are controlled. Genetic regulation.
genetic regulation or regulation of gene expression covers the entire process for transcription of the genetic code in the nucleus of the formation of proteins in the cytoplasm. Regulation of the gene expression provides all living organisms with the ability to respond to change in their environment. In animals that have many different types of cells, tissues, and organs, differential regulation of gene expression also permits the different cell type in the body to each perform their specialized function. Although a cardiac myocyte contains the same genetic code as the renal tuber epithelial cell, many genes are expressed in cardiac cells that are not expressed in the renal tubular cells. The ultimate measure of gene the expression is whether and low how much of the gene products proteins are produced because proteins carry out cell function specified by the genes. Regulation of the gene expression can occur at any point in the pathway.
Regulation, genetic regulation, or regulation of gene expression converts the entire process from transcript of the genetic code in the nucleus to the formation of proteins in the cytoplasm. Regulation of gene expression provides all living organisms with the ability to respond to changes in their environment. In animals, they have many different types of cell tissues. Organ differential regulation of gene expression also permits the different types of cell in the body to each perform their specialized functions. All through the cardiac myocyte cell, many genes are exposed in cardiac cells that are not expressed in renal tubular cell. The ultimate measure of products proteins are produced because proteins carry out cell functions specified by their genes. Regulation of gene expression can occur at any point in the pathway of transcription, DNA processing, and translation. The ultimate measure of products proteins are produced because proteins carry out the cell function specified by the gene. Regulation of gene expression can occur at any point in the pathways of transcript, RNA processing, and translation. The promoter control gene expression, synthesis of cellular proteins in a complex process that starts with transcription of DNA into RNA. Transcription of DNA controlled by regulatory elements found in the promoter of a gene. In eukaryotes, which includes all mammals, the basal promoter consists of a sequence of bases, TATA, called TATA box. The binding state for the TATA binding protein and several other important transcription factors that are called collectively referred to as the transcription factor IID complex. In addition to the transcription factor IID complex, this region is where transcription factor IIB binds to both the DNA and the RNA polymerase to facilitate transcription of the DNA into RNA. This basal promoter is found in all protein genes, and polymerase must be built with a basal promoter before it can be traveling along the DNA strand to synthesize RNA. The upstream promoter.
Upstream promoter is located further upstream from the transcription start site and contains several bindings sites for positive or negative transcription factors that can affect the transcription through interaction with proteins bound to the basal promoter. The structure and the transcription factor bind sites in the upstream promoter vary from gene to give rise to the different expression patterns of genes in different tissues. Transcription of the genes is eukaryocyte is also influenced by enhanced of DNA that can bind transcription factors. Enhancers can be located a great distance from the gene. They act on even on a different chromosome. They can also be located upstream or downstream to the gene that they regulate. Although the enhancers may be located far from their target gene, they may be relatively rapidly metabolize with their energy used for replenishment of ATP storage. Thus, the CMP acts as an enzyme activator for the enzyme phosphorylase and thereby helps control the intercellular ATP concentration. Another interesting example of both enzyme inhibition and enzyme activation occurs in the formation of the purines and pyrimidines. These substances are needed by the cell in approximately equal quantities for the formation of DNA and RNA. When purines are formed, they inhibit the enzymes that are required for formation of additional purines. However, they are activated the enzyme for formation of pyrimidines. Conversely, the pyrimidines inhibit their own enzyme but activated the purine enzymes. In this way, there is the continual crosstalk between the synthesizing systems for these two substances, resulting in almost exactly equal almost of the two substances in the cell at all times. Summary: there are two principal mechanisms whereby cell control proper proportion and quantities of different cellular constituents. Genetic regulation, enzyme regulation. Genes can be activated or inhibited, and likewise, the enzyme system can be activated or inhibited. These regulatory mechanisms usually function as feedback control system and they continually monitor the cell biochemical composition and make correction as needed. However, on occasion, substance from outside the cell, especially some of the hormones discussed in the text, also control the intercellular biochemical reaction by inactivating or inhibiting one or more into the intercellular control system. DNA genetic system controls cell reproduction. Cell reproduction is another example of the key evacuation role that the DNA genetic system plays in all life processes. Genes and their regulatory mechanism determine cell growth characteristic and when or whether cells will divide to form new cells. In this way, the all important genetic system control each stage in the development of the human, from the single cell fertilized ovum to the whole functioning body. Thus, if there is any central theme of life, it is the DNA genetic system. Life cycle of the cell. Life cycle of the cell is period from cell reproduction to the next cell reproduction. When mammalian cells are not inhibited and the reproducing cell rapidly as they can, this life cycle may be as little as 10 to 30 hours. It is terminated by a series of distinct physical events called the mitosis, thus cause the division of the cell into two new daughter cells. The events of mitosis are shown in figure 314 described later. The actual stage of mitosis, however, lasts for only about 30 minutes and thus more than 95% of the life cycle of even rapidly reproducing cells is represented by the internal between the mitosis cell interface. Except is the special condition of the rapid cellular reproduction, inhibitory factors almost always stop uninhibited life cycle of the cell. Therefore, different cells of the body actually have life cycle periods of the very form of little as 10 hours for highly stimulated bone marrow cell to entire lifetime of the human body of the many nerve cells. Cell reproduction begins with replication of DNA. First step of the cell production is replication and duplication of all DNA in this chromosome. It is only after this replication has occurred that mitosis can take place. DNA begins to duplicate 5 to 10 hours before mitosis, and the duplication is completely in 4 to 8 hours. The net result is two exact replicates of all DNA. These replicas become the DNA in the two new daughter cells and they will be formed at mitosis. After replication of the DNA, there is another period of 1 to 2 hours before mitosis begins abruptly. Even during the period, preliminary changes that will lead to the mitotic process are beginning to take place. DNA is replicated in much the same way the RNA is transcribed from DNA, except for a few important differences. Both strands of the DNA in each chromosome are replicated, not just one of them.
Both entire
কথা
both entire strands of DNA helix and replicated from end to end rather than a small portion of them, as occurs in the transcription of RNA. Multiple enzymes called DNA polymerase, which is comparable to RNA polymerase, are essential for replication DNA. DNA polymerase attaches and moves along the DNA template strand, adding a new nucleotides in 5 to 3 direction. Another enzyme, DNA ligase, cause bonding of successive DNA nucleotides, and in the end another using high energy photon bands to energize these attachments. Replication fork formation. Before DNA can be replicated, the double strand molecules must be unzipped into two single strands. Because the DNA helix in each chromosome are approximately 6 centimeters in length and have millions of helical turns, it would be impossible for the two newly formed DNA helix to uncoil from each other were it not for some special mechanism. This uncoiling is achieved by DNA helicase enzyme that break the hydrogen bond between the base pairs of DNA, permitting the two strands to separate into a Y shape known as replication fork. The area that will be the template for replication to begin. DNA is directional in both strands, signified by a 5 to 3 and figure. See figure 3 to 15. Replication process only in the 5 to 3 direction. At the replication fork one strand, the leading strand is oriented in the 3 to 5 direction towards the replication fork, while the lagging strand is oriented 5 to 3 away from replication fork. Because of different orientations, the two strands are replicated differently. Permanent binding. Once the DNA strands have been separated, a short piece of RNA called RNA prime binds to 3 and end of the leading strand. Primes are generated by enzyme DNA primase. Primes always bind as the starting point for DNA replication. Elongation. DNA polymerase are responsible for creating the new strand by a process called elongation. Because replication process proceeds in the 5 to 3 direction to the leading strand, the newly formed direction is continuous. The lagging strand begins replication by binding its multiple primes that are only several bases apart. DNA polymerase then adds pieces to DNA called Okazaki fragments to the strands between primes. The process of replication is discontinuous because the newly created Okazaki fragments are not yet connected. An enzyme, DNA ligase, joins the Okazaki fragment to form a single fine unfolded strand. Termination after the continuation and discontinuous strands are both formed. The enzyme exonuclease removes the RNA primes from the original strand, and the primes are replaced with appropriate bases. Another exonuclease profits the newly formed DNA, checking and clipping off any mismatched and unpaired residues. Another enzyme, topoisomerase, can transiently break the phosphodiester bond in the backbone of DNA strand to prevent the depleted the parent strand and its complementary DNA strand coils into the double helix shape. The process of replication therefore proceeds two DNA molecules, each with one strand from the parent DNA and one new strand. For this reason, DNA replication often described as semi-conservative. Half of the chain is part of the original DNA molecule and half is brand new DNA repair, DNA proofreading, and mutation. During the hour of between DNA replication and
Beginning of the mitosis, there is period of active repair and proofreading of the DNA strands. Wherever inappropriate DNA nucleotides have been matched up with the nucleotides of the original template strand, special enzymes cut out of the defective areas and replace them with appropriate complementary nucleotides. This repair process, which is achieved by the same DNA polymerase and DNA ligases that are used in replication, it is referred to as DNA proofreading. Because of repair and proofreading, mistakes are rarely made in the DNA replication process. When a mistake is made, it is called a mutation. The mutation may cause formation of some abnormal protein in the cell rather than a needed protein, which may lead to abnormal cellular function and sometimes even cell death. Given that many thousands of genes exist in the human genome and that the period from one human generation to another is about 30 years, one would expect as many as 10 or many more mutation is the passage of the genome from the parent of to offspring. As a further protection, however, each human genome is represented by two separate sets of chromosomes, one derived from each parent with almost identical genes. Therefore, one functional gene of each pair is almost always available to the child despite mutation, chromosome and their replication. The DNA helix of the nucleus are packed in chromosome. Then human cells contain 46 chromosomes arranged in 23 pairs. Most of the genes in the two chromosomes of each pair are identical or almost identical to each other, so it is usually stated that the different genes also exist in pairs, although occasionally this is not the case. In addition to DNA, there is a large amount of protein in the chromosome, composed mainly of many small molecules of electrophosphativity charged histones. The histones are organized into vast numbers of small bovine-like cores. Small segments of each DNA helix are coiled sequentially around one core after another. The histone cores play an important role in regulation of DNA activity because as long as the DNA is packaged tightly, it cannot function as a template for formation of RNA or replication of new DNA. Furthermore, some of regulatory proteins decontains the histone packaging of the DNA and allows some segments at a time from RNA. Several non-histone proteins are also major components of chromosome, functioning as chromosomal structure proteins and its connection with an genetic regulatory machinery as activators, inhibitors, and enzymes. Replication of the chromosome in the entirely occurs during the next few minutes after the replication of DNA helix has been completed. The new DNA helix collect new protein molecules as needed. The two newly formed chromosomes remain attached to each other until time for mitosis at a point called of centromere located near their center. These duplicated but still attached chromosomes are called the chromatid. Cell mitosis. Actual process whereby the cell splits into two new cells is called the mitosis. Each chromosome has been replicated to form the two chromatids. Mitosis follows automatically within 1 to 2 hours in many cells. Mitotic apparatus. Function of centrioles. One of the first events of the mitosis takes place in the cytoplasm in or around the small structure called the centrioles. During the later part of the interphase, as shown in figure 3 to 14, two pairs of centrioles lie close to each other near one pole of the nucleus. These centrioles, like the DNA and chromosomes, are also replicated during the interphase, usually shortly before replication of DNA. Each centriole is a small cylindrical body about 0.4 micrometer long and about 0.15 micrometer in diameter, consisting mainly of 9 parallel tubular structures arranged in the form of a cylinder. The two centrioles of each pair lie at the right angles to each other. Each pair of the centrioles, along with attached pericentriole material, is called the centrosome. Shortly before mitosis takes place, the two pair of centrioles begin to move apart from each other. This movement is caused by the polymerization of protein molecules growing between the respective centrioles pairs and actually pushing them apart. At the same time, the other microtubules grow rapidly away from each of the centriole pairs, forming a spinning star called the ester in each end of the cell. Some of the spines of the star penetrate of nuclear membrane and help separate the two sets of the chromatids during mitosis. The complex of microtubules extending between the two new centriole pair is called the spindle, and the entire set of microtubules pulse the two pairs of centrioles is called the mitotic apparatus. Prophase. The first stage of mitosis is called the prophase, is shown in figure 3 to 14 A, B, and C. While the spindle is forming, chromosomes of the nucleus, which is in the interphase consists of loosely coiled strands, become condensed into well-defined microsomes. Prometaphase. During the prometaphase stage, the growing microtubules spines of the stars fragments of the nuclear envelope. At the same time, the multiple microtubules from the star attach to the chromatids at the centromeres, where the paired chromatids are still bound to each other. The tubules then pull one chromatid of each pair toward one cellular pole and its partner toward the opposite pole. Metaphase. During the metaphase stage, the two esters of the mitotic apparatus are pushed further apart. This pushing is believed to occur because the microtubule spines form two esters where they integrate with each other to form the mitotic spindles, push each other away. Minute contractile protein molecules called the molecule motors, which may be composed of the muscle protein acting, extend between the respective spines and using a stepping action as the muscle actively side the spines in a reverse direction along each other. Simultaneously, the chromatids are pulled tight by their attached microtubules to their center of the cells, lining up to form the equatorial plate of the mitotic spindle. Anaphase. During the anaphase stage, the two chromatins of each chromosome are pulled apart at the centromere. All 46 pairs of chromatids are separated from two separate sets of 46 daughter chromosomes. One of these sets is pulled toward one mitotic ester and other is pulled towards the other ester as the two respective poles of the dividing cell are pushed still further apart. Telophase. In the telophase stage, the two sets of daughter chromosomes are pushed completely apart. Then the mitotic apparatus disappears and a new nuclear membrane develops around each set of the chromosomes. The membrane is formed from portion of the endoplasmic reticulum that are already present in the cytoplasm. Shortly thereafter, the cell pinched into midway between the two nuclei. The pinching is caused by the formation of the contractile ring of the microfilaments composed of action and probably myosin, two contractile proteins of muscle at the junction of the newly developing cells that pinch them off from each other.
Control of the cell growth and cell reproduction. Some cell grow and reproduce all the time, such as blood forming cells of the bone marrow, the germinal layers of the skin, and the epithelial of the gut. Many other cells, however, such as smooth muscle cells, may not reproduce for many years. A few cells, such as the neurons or the most strained muscle cells, do not reproduce during the entire life of a person, except during original original period of fetal life. In certain tissues, insufficiency of some type of cell causes them to grow and reproduce rapidly until appropriate number of these cells are again available. For example, in some young animals, even 8 years, 7 or 8 years of 8 years of the liver can be removed. 7/8 of the liver can be removed surgically, and the cell of the remaining 1/8 will grow and divide until the liver mass returns to almost normal. The same phenomenon occurs for many glandular cells and most cells of the bone marrow, subsequentous tissue, essential epithelium, and at most any other tissue except highly differentiated cells such as nerves and muscle cells. The mechanism that maintain proper number of the different types of cells in the body are still poorly understood. However, experiments have shown at least 3 ways in which growth can be controlled. First, growth often is controlled by growth factors that come from one part of the body. Some of these growth factors circulate in blood, but others originate in adjacent tissue. For example, the epithelial cell of some glands, such as the pancreas, fail to grow without a growth factor from the underlying connective tissue of the gland. Second, most normal cells stop growing when they run out of space to grow. The phenomenon occurs when cells are grown in tissue culture. The cell grow until they contact a solid object, and then growth stops. Third, cell growth in tissue culture often stops growing when minute amounts of their own secretion are allowed to control in the culture medium. This mechanism too could provide a means for negative feedback control of growth. Telomeres prevent the degradation of chromosomes. A telomere is a region of primitive nucleotide sequence located at each end of a chromatid. Telomeres serve as protective caps that prevent the chromosome from deterioration during cell division. During cell division, a short piece of primer DNA attaches to the DNA strand to start the replication. However, because of the primer does not attach at the very end of the DNA strand, the copy is missing a small section of DNA. With each cell division, the copied DNA loses additional nucleotides from the telomere region. The nucleotide sequence provided by the telomere therefore prevents the degradation of genes near the end of chromosomes. Without telomeres, the genomes would progressively lose information and be tracted after each cell and be tracted each cell division. Thus the telomeres can be considered to be disposable chromosomal buffers that help maintain stability of the genes but are gradually consumed during repeated cell division. Telomere enzyme. Figure 3 to 16. Control of cell reproduction by telomeres. And telomeres, the cell chromosomes, are copied by telomeres which, in the absence of telomere activity, shorten with each cell division until the cell stops replicating. Therefore, most cells of the body cannot replicate indefinitely. In cancer cells, telomeres in an activated and telomere length is maintained so that the cell continue to replicate themselves uncontrollably. Each time a cell divides, an average person loses 30 to 200 base pairs from the end of cell telomeres. In human blood cell, The length of telomeres range from 8,000 base pairs to birth to as low as 1,500 in older people. Eventually, when the telomeres shorten a critical length, the chromosomes become unstable, and the cell die. This process of telomere shortening is believed to be an important reason for some of the physiological changes associated with aging. Telomere erosion can also occur as a result of disease, especially those associated with oxidative stress and inflammation. In some cells, such as stem cells of the bone marrow or skin that must be replenished throughout life, a germ cell in the ovaries and testes, the enzyme telomerase adds bases to the ends of telomeres so that many more many more generations of cells can be produced. However, telomerase activity is usually low on most cells of the body, and after many generations, the descendant cells will inherit defective chromosomes because sene- senescence and case diving, limiting cell proliferation and maintaining gene stability in cancer cells. Telomerase activity is abnormal activity, so the telomerase length is maintained, making it possible for the cells to replicate over again uncontrollably. Some scientists have therefore proceeded that telomerase shortening protects us from cancer and other proliferative disease. Regulation of cell size.
Of cell size. Cell size is determined almost entirely by the amount of functioning DNA in the nucleus. If replication of DNA does not occur, the cell grows to a certain size and thereafter remains at this size. Conversely, use of the chemical collagen makes it possible to prevent the formation of the mitotic spindles and therefore prevent mitosis, even though the replication of the DNA continuous. In this event, the molecule contains fat greater quantities of DNA than the normally does, and the cell grows proportionally larger. It is assumed that this cell growth results from increased production of RNA and cell proteins, which in turn cause the cell to grow larger. Cell differentiation. A special characteristic of cell growth and cell division is cell differentiation, which refers to change in the physical and functional properties of the cells as they proliferate the embryo to form the different body structure and the organs. The following description of an especially interesting experiment helps explain this process. When the nucleus from an intestinal mucosal cell of a frog is surgically implanted into a frog ovum from which the original ovum nucleus was removed, the result is often the formation of a normal frog. These experiments demonstrated that even the intestinal mucous cell, which is a well differentiated cell, carries all the necessary genetic information for development of the structure required in the frog's body. Therefore, it has become clear that different result not from loss of genes but from the selective representation of different gene promoters. In fact, electron microscope suggests that some segments of DNA helices that are wound around the histone course because so condensed that they no longer uncoil to form the RNA molecules. One explanation for this is as follows. It has been supposed that the cellular genome begins at a certain stage of cell differentiation to regulatory protein that forever after represses a select group of genes. Therefore, the repressed genes never function again, regardless of the mechanism. Mature human cell each production a maximum of about 80,000 to 10,000 proteins rather than the potential 20,000 to 25,000 or more that would be produced if all genes were active. Embryologically, experiments have shown that certain cells in an embryo control differentiation of adjacent cells. For example, the primordial Codamesodum is called the primary organization of the embryo because it forms a focus around which the remainder cell light jalao na.
Embryological experiments have shown that certain cells into the embryo control differentiation of adjacent cells. For example, the primordial chromosoderm is called the primary organizer of the embryo because it forms a focus around which the remainder of the cell embryo develops. It differentiates into a mesodermal axis that contains segmentally arranged somates, and as a result of induction in the surrounding tissue, causes the formation of essentially all the organs of the body. Another instance of induction occurs when the developing eye vesicles come in contact with the ectoderm of the head and cause the ectoderm to thicken into a lens plate and folds inwards to form the lens of the eye. Therefore, a large share of embryo develops as a result of such inductions, with one part of the body affecting another part and this part affecting still other parts. Thus, although our understanding of cell differentiation is still hazy, we are aware of many control mechanisms thereby differentiation could occur. Apoptosis programmed called death. In many trillions of body cells are members of a highly organized committee in which the total number of cells is regulated not only by controlling the rate of cell division but also by controlling the rate of cell death. When cells are no longer needed or become a threat to the organism, they undergo a suicidal program called death or apoptosis. This process involves a specific proteolytic cascade that causes the cell to shrink and condense, disassemble its cytoskeleton, and alter its cell surface so that a neighboring phagocyte cell, such as macrophage, can attach to the cell membrane and digest the cell. In contrast to programmed death, cells that die as a result of acute injury usually swells and bursts due to loss of cell membrane integrity, a process called cell necrosis. Necrotic cell may spill other contents, causing inflammation and injury to neighboring cells. Apoptosis, however, is an orderly cell death that results in disassembly and phagocytosis of the cell before the leakage of the content occurs. The neighboring cells usually remain healthy. Cancer. Apoptosis is inherited by activation of a family protease and called caspases, which are enzymes that are synthesized and stored in the cell as inactive procaspases. The mechanism of activation that caspases are complex, but once activated, the enzymes cleave and activate other procaspases, triggering a cascade of rapid breakdown proteins within the cell. The cell thus dismantles itself and it remains are rapidly digested by neighboring phagocytic cell. A tremendous amount of apoptosis occurs in tissues that are being remodeled during development. Even in adult humans, billions of cells die each hour in tissue such as the intestine and bone marrow and are replaced by new cells. Programmed cell death, however, is normally balanced by formation of new cells in healthy adults. Otherwise, the body's tissues would shrink and grow excessively. Abnormalities of apoptosis may play a key role in the neurodegenerative disease such as Alzheimer's disease, as well as in cancer and autoimmune disorder. Some drugs that have been used successfully for chemotherapy appear to induce apoptosis in cell cancer. Cancer. Cancer may be caused by mutation or by some other abnormal activation of cellular genes that that control cell growth and cell mitosis. Proto-oncogenesis are normal genes that code for various proteins that control cell division. If mutated or excessively activated, proto-oncogenes can become abnormally functioning oncogenes capable of causing cancer. As many as 100 different oncogenes have been discovered in human cancers. Also present in all cells are anti are an antioncogens, also called tumor suppressor genes, which suppress the activation of a specific organism oncogenes. Therefore, loss or inactivation of oncogenes can allow activation of oncogenes that lead to cancer. For several reasons, only a minute fraction of the cell that matures in the body ever leads to cancer. First, most mature cells have less survival capability than normal cells, and they simply die. Second, only a few of the matured cells that survive become cancerous because most mutated cells still have normal feedback controls that prevent excessive growth. Third, cells that are potentially cancerous are often are often destroyed in the body immune system before they grow into a cancer. Most mutated cells from abnormal proteins within the cell bodies because of their altered genes, and these proteins activate the body's immune system, causing it to form antibodies or sensitized lymphocytes that react against the cancerous cells, destroying them. In people whose immune system have been suppressed, such as in persons taking immunosuppressant drugs after kidney or heart transplant, probably probably that a cancer will develop in multiple as much as fivefold. For the simultaneous presence of several different activated oncogenes is usually required to cause a cancer. For example, one such gene might promote rapid production of cell line, but no cancer occurs because another mutant gene is not present simultaneously to form the needed blood vessel. What is the cause of altered genes? Considering the many trillions of new cells are formed each year in humans, a better question might be asked why all of the cells do not develop millions or billions of mutinous cancerous cells. The answer is the incredible precision with which DNA chromosome strands are replicated in each cell before mitosis can take place, along with the proofreading process that cuts and repairs many abnormal DNA strands before the mitosis process is allowed to proceed. Yet, despite these inherited cellular precautions, probably one newly formed cell in every few million still has significant mutation characteristics. Thus, this alone is all that is required for mutations to take place. So we can suppose that a large number of cancer cells are merely the result of an unlucky occurrence. However, the probability of mutations can be generally increased when a person is exposed to certain chemicals, physical or biological factors, including the following: ionizing radiation such as X-rays, gamma rays, particle radiation from the radioactive substance, and even ultraviolet light can predispose individual individuals to cancer. Ions from in tissue cells under the influence of such radiation are highly reactive and can rupture DNA strands, causing many mutations. Chemical substances of certain types may also cause mutations. It was also discovered long ago that various aniline derivatives that are likely to cause cancer, and thus works in chemical plants producing such substances, if unprotected, have a special predisposition to cancer. Chemical substances that can cause mutation are called can carcinogens. The carcinogens that currently cause the greatest number of deaths are those in cigarette smoke. The carcinogens cause over 30% of all cancer deaths and at least 85% of lung cancer deaths. Physical irritations can also lead to cancer, such as continued abrasion of the lines of the intestinal tract by some type of food. The damages of these tissues lead to rapid mitotic replacement of the cells. The more rapid the mitosis, the greater the chance of mutation. Heredity tendency to cancer occurs in some families. The heredity tendency results from the fact that most cancer require not only mutation but two or more mutations before cancer occurs. In families that are particularly predisposed to cancer, it is presumed that one or more cancerous genes are already mutated in the inherited genome. Therefore, far fewer additional mutations must take place in such family members before a cancer begins to grow. Certain types of oncoviruses can cause various types of cancer. Some examples of viruses associated with cancer include human papillomavirus, HPV, hepatitis B, and hepatitis C virus. Epstein-Barr virus, human immunodeficiency virus, HIV, human T-cell leukemia virus, Kaposi sarcoma, associated herpes virus, KSHV, and Merkel cell polymerase virus. Although the mechanisms whereby oncoviruses cause cancer are not fully understood, there are at least two potential ways. In the case of DNA viruses, the DNA strand of the virus can insert itself directly into one of the chromosomes, thereby causing a mutation that leads to cancer. In the case of RNA virus, some of these viruses carry with them an enzyme called reverse transcriptase that causes DNA to be transcripted from the RNA. The transcribed DNA then inserts itself into the animal cell genome, leading to cancer. Invasive characteristics of cancer cell. The major difference between a cancer cell and a normal cell are as follows. The cancer cell does not respect usual cellular growth limits because these cells do not require all the same growth factors that are necessary to cause the growth of normal cells. Cancer cells are often far less adhesive to one another than a normal cell. Therefore, they tend to wander through the tissue, enter blood stream, and be transported all through the body, where they form needy from numerous new cancerous growths. Needy or non-invasive diagnostic imaging. Non-invasive diagnostic imaging and Merkel cells polymerase. Although the mechanism whereby oncoviruses cause cancer are not fully understood, there are at least two potential ways. In the case of DNA viruses, the DNA strand of the virus can insert itself directly into one of the chromosomes, thereby causing a mutation that leads to cancer. In the case of RNA virus, some other viruses carry with them an enzyme called reverse transcription that causes DNA to be transcribed from the RNA. The transcribed DNA then inserts itself into the animal cell genome, leading to cancer. Some cancer also produce angiogenic factors that cause many new blood vessels to grow angiogenic grow into the cancer, thus supplying the nutrition required for cancer growth. Why do cancer cells kill? Tissue causes compete with normal tissue. Cancer tissue competes with normal tissue for nutrition because cancer cells continue to proliferate indefinitely with their numbers multiplying every day. Cancer cells soon demands essential all the nutrition available to the body or to essential parts of the body. As a result, normal tissue gradually sustain nutritive death. Cancers cause disruption of vital organ functions. For example, a lung cancer might replace healthy tissue to the extent that the lung cannot absorb enough oxygen to maintain tissue in the rest of the body.